Environmental contamination begins with bioaccumulation. Toxicants enter aquatic environments, bind to microscopic particulates, and are ingested by base-level organisms like zooplankton. This USGS diagram illustrates how these toxicants biomagnify as they move through the food web. Because these chemicals resist metabolic degradation, their concentrations multiply exponentially at each trophic level, delivering the heaviest chemical burdens to apex predators. Historically, risk assessment relied on the LD50 metric, the acute dose required to kill exactly half of a test population. However, many modern contaminants are endocrine-disrupting chemicals, or EDCs, which rarely cause immediate mortality upon exposure. The risk is sublethal. These compounds possess chemical structures that mimic, block, or alter the body's endogenous hormones, effectively reprogramming developmental and reproductive trajectories. Birds are uniquely vulnerable to this process. Their specialized reproductive cycles and seasonal metabolic demands create a physiological substrate that cannot be accurately predicted by mammalian testing models alone. Predicting these systemic risks requires looking past simple mortality curves to trace the entire adverse outcome pathway, mapping the damage from an initial molecular binding event to the eventual decline of the population. Every adverse outcome pathway begins at the molecular level, where the exact geometry of a chemical dictates its toxic potential. Consider polychlorinated biphenyls, also known as PCBs. Their toxicity depends on the precise positioning of chlorine atoms on the molecule's rings. This chart compares specific structures to determine their relative threat. PCB 126 at the top has a chlorine arrangement facilitating direct interaction with receptors. Directly below it is TCDD, a dioxin serving as the baseline standard for comparing toxicity. This structural comparison yields a toxic equivalency factor, or TEQ. By measuring an EDC's similarity to dioxin, researchers calculate specific values used to quantify the risk of complex chemical mixtures found in the wild. That molecular geometry dictates cellular infiltration. Because many EDCs are lipophilic, they pass smoothly through the cell's lipid bilayer into the cytoplasm. This pathway diagram illustrates the aryl hydrocarbon receptor signaling process. The exogenous ligand structure determines its affinity for binding to the AHR complex waiting within the cell. Binding triggers an immediate consequence. The receptor complex sheds its chaperone proteins and translocates across the nuclear membrane. Inside the nucleus, the complex dimerizes with the AARNT protein and binds to the xenobiotic response element on the DNA strand. The ultimate result is increased transcription of CYP1 mRNA, which initiates a cascade of oxidative stress and altered metabolism. Other EDCs, like the pesticide metabolite DDE, bypass this pathway to act as direct xenoestrogens or androgen blockers by hijacking the cell's steroid receptors. This microscopic receptor binding event acts as the irreversible trigger for all subsequent physiological dysfunction in the bird. While cellular mechanics are universal, avian life history traits create unique vulnerabilities to these molecular triggers. Lipophilic EDCs bioaccumulate in the fat reserves of female birds over their lifetimes. The extreme metabolic demands of egg production force females to mobilize these lipid reserves, partitioning massive chemical burdens directly into the yolk of the developing egg. Avian embryos effectively stew in a concentrated EDC mixture during their most critical windows of sexual and neurological differentiation. The timing and severity of this damage depend on whether a species utilizes a precocial or ultrocial developmental strategy. Regulatory tests focus on precocial birds, like the Japanese quail. Because they hatch highly developed, their reproductive axes are organized primarily in ovo. In contrast, ultricial species develop many steroid-dependent neural systems post-hatch, extending their window of vulnerability long after they leave the egg. Mapping an EDC's impact involves correlating its molecular mechanism with the specific time-sensitive developmental trajectory of the target species. As a bird matures, these cascading molecular failures begin to collapse entire organ systems. The Deepwater Horizon oil spill serves as a real-world model for complex, multi-system exposure to polycyclic aromatic hydrocarbons, or PAHs. This influence diagram maps the potential routes of exposure for birds in contaminated environments, specifically through the ingestion and absorption of oil. Exposure triggers phase 1 PAH conversion via the CYP enzymes we mapped earlier, resulting in altered gene expression and systemic oxidative stress. These molecular disruptions lead directly to secondary clinical indicators, including elevated liver enzymes and widespread inflammation. In the final stage of deterioration, clinical indicators manifest as catastrophic organ and tissue effects, including hemolytic anemia and total reproductive failure. These internal failures are worsened by the bird's behavioral response to the contamination. A seabird slicked with oil will instinctively begin mechanical preening to restore the buoyancy and insulation of its feathers. This behavior inadvertently creates a feedback loop of toxic ingestion, compounding the initial adrenal and metabolic dysfunction already occurring inside the bird. A singular molecular binding event expands into a multi-system failure that destroys flight efficiency, homeostasis, and reproductive viability. Designing regulatory frameworks to protect these ecosystems requires testing protocols that account for this multi-layered physiological complexity. A single environmental chemical can act simultaneously as an estrogen agonist, an androgen antagonist, and a thyroid disruptor. This tiered screening strategy used by the EPA and OECD begins with in vitro assays to identify early endocrine activity at the receptor level. However, isolated cellular data must be mapped against whole organism physiological axes using in vivo assays. Multi-generational tests, such as the Japanese quail 2-generation test, are essential to capture the delayed effects of in ovo exposure on adult reproductive fitness. This rigorous, multi-tiered approach provides the data necessary for the broader One Health framework, linking wildlife health directly to the health of the ecosystem. Protecting birds means moving beyond the search for simple acute toxicity and instead mapping the full adverse outcome pathway from the single molecular receptor to the decline of the species.